everyone, and thank you so much for the opportunity of presenting my PhD project today. I will present to you the validation of exportin 1 and mitogen-activated protein kinase 2 as molecular drug targets for ALS. As you all know, effective therapeutic options are lacking in ALS, and better characterization of disease mechanism could help with coming up with new strategies for treatment of ALS. Therefore, our project aimed to validate the molecular candidates which with potential implication in ALS pathology we derived from the MAXIMOD project. In the context of this consortium, we performed multi-omic analysis of postmortem tissues, prefrontal cortex for postmortem tissues from ALS patients and control, as well as 4 different mouse models of the disease, in order to characterize early and sex-specific disease mechanism in ALS, and with different integrational analyses of multi-omics in human and mouse samples, we identified interesting and most promising dysregulated pathways and targets, which more details on this project will be tomorrow presented in session 3 by my colleague Dr. Zeplev. But the focus of my PhD project was to validate these selected molecular targets of this, from this data in a stress-induced in vitro model. So while we observed transcriptional differences and also cell composition differences between different mouse models and ALS human patients, we observed that a MAP kinase pathway was consistently altered in different models, which highlighted the importance of this pathway in the pathomechanism of ALS. So we focused on MEC2, which one of the most important components of this pathway. It's involved in RAS-RAF-MEC-ERK pathway, which is involved in really important cellular functions like cell proliferation and survival, and it's the main regulator of ERK phosphorylation. For the next target, we also observed that nucleocytoplasmic transport was altered in enrichment analysis, and exportin 1 is also an important component of this pathway, which is a mediator of nucleotransport of many important proteins and RNAs from nucleus to the cytoplasm. And it was also highly upregulated in both protein and gene level in different mouse models. So we decided to further validate these 2 candidates. In order to assess the role of these targets in neural survival, we decided to we tried to establish a primary cortical culture which is intoxicated to mimic some of the well-known pathological pathways in ALS. As you all know, glutamate excitotoxicity is well known in ALS pathology, and increased synaptic glutamate results in dysregulation of mitochondrial function, and at the end it's a result into cell death. So with 5 millimolar glutamate for 6 hours, we induced apoptosis in this primary cell culture, and we stained for Cliff caspase 3, which is an apoptotic marker. And we observed that this glutamate induction results in increased cell death, and also it significantly reduced the average neurite length. For the other model, we decided to induce stress granules. We used sodium arsenate for 3 hours to induce stress granules, which we visualized it with staining for G3BP1, which is a stress granules marker. As you know, stress granules dysregulation is also known to be associated with different aspects of ALS pathology. For example, it is hypothesized to be act as a seeding mechanism for aggregation of some proteins like TDP43, as well as it associates with mitochondrial dysfunction, as well as inflammation and nucleocytoplasmic transport deficit. So induction of stress granules also resulted in a significant increase in cell death, as well as reduction in neurite length. So in order to validate the neuroprotective effect of these targets, we decided to modulate their expression and their activity by already available FDA-approved small molecule inhibitors. So teramitinib is a reversible allosteric inhibitor of MEC1 and 2, and it basically go and stalk to the activation loop of MEK, which then results in a reduction of phosphorylation of ERK. It also abrogates the feedback loop of the cascade, and it induces phosphorylation of MEK, which we actually showed that with Western blot. So we treated the cells for 72 hours with different concentration of teramitinib to 2 to 200, and then we did the Western blot, and we observed that teramitinib significantly reduced phosphorylation of ERK. The total protein expression of ERK and MEK didn't change significantly, but it also seemed to induce phosphorylation of MEK. And this way we showed the interaction of drug and target. So we wanted to see how glutamate excitotoxicity is affecting this cascade and changes the phosphorylation of protein level of MEK and ERK. So we observed that so we treated the cells with different concentration of teramitinib for 72 hours, and at day in vitro 7 we applied glutamate or iodine-meso, and we observed that excitotoxicity itself significantly increased the phosphorylation of ERK, which then teramitinib was able to completely restore this highly phosphorylated ERK. And we observed that the MEK itself didn't change with the teramitinib, and also glutamate excitotoxicity didn't change it, and teramitinib also induced phosphorylation of MEK. So we wanted to see how this inhibition if it has any neuroprotective effect. So we treated the cells with different concentration, and we also applied the stressor, the glutamate, and we checked for the cell death with analyzing Cliff caspase positive cells, and also with measuring neurite length, and we observed that teramitinib 220 and 200 nanomolar teramitinib for 72 hours significantly reduced the cell death, the number of Cliff caspase 3 positive cells, and also it induced neurite regeneration. So we wanted to test the same effect also in the stress-induced model, stress granule-induced model, and we observed that the same as glutamate, we saw that sodium arsenate increased phosphorylation of ERK, which was again restored by teramitinib. MEK2 didn't change, but phosphorylation of MEK was also induced by applying teramitinib. To check if it has any neuroprotective effect in this model, we first checked if applying teramitinib is changing the intensity of stress granules. We observed that it's not significantly altered stress granules intensity, or like G3BP1 signal intensity, but we observed that all 3 concentration of teramitinib increased significantly the average neurite length in this model. So coming to our next target, we talked about exportin-1, which has some targets. The targets bind to the exportin-1, and they make this RAN-GTP complex, and then it's able to translocate from the nucleus to the cytoplasm. Selinexor is also an FDA-approved small molecule inhibitor, which is now at the moment is being used in clinical trial for cancer, and this binds to exportin-1 and inhibits its translocation to the nucleus. So we wanted to show how this drug target interaction. We hypothesized that with inhibiting of exportin-1, then it's going to result into accumulation of its target into the nucleus. So we chose P62, which is an autophagy receptor and one of the common targets of exportin-1. And first, I showed that with ICC. So I treated the cells with different concentration of selinexor, and you can see the higher concentration of selinexor resulted into accumulation of P62 signal in the nucleus. For further showing this, I wanted to see, I treated the cells and did the Western blot for subcellular fractionation. So I wanted to see this individually in cytoplasm and nuclear fractions. And as you can see, I also checked for the exportin-1, and exportin-1 protein level was significantly increased both in cytoplasm and nuclear fractions, which kind of showing the compensatory mechanism of the cell, but then increase in accumulation of P62 was more pronounced in nuclear fractions. So in order to see if there is any neuroprotective effect by inhibition of exportin 1 by selinexor, we checked again and we stained for cliff caspase 3 positive cells, and you can see that 1 and 10 nanomolar selinexor significantly reduced apoptotic cells. And how we can explain this as a mechanism of action behind it, we know that the excitotoxic level of glutamate induces the nuclear egress of several pathological proteins like TDP-43, and then by inhibition of transmission of these protein into the cytoplasm, then maybe we can see this neuroprotective effect. And this is what at the moment we are investigating. I want to show the accumulation of TDP-43 in the nuclear fractions. And I also checked the neuroprotective effect of exportin 1 inhibition in a stress granules toxicity model. And I observed that, again, 1 nanomolar and 10 nanomolar selinexor significantly reduced apoptosis, but it didn't alter the stress granules intensity. So to summarize, the multi-omic analysis in ALS-affected brains identified exportin 1 and MEK2 as molecular drug targets for ALS. We modulated the expression of these two targets with pharmacological small molecule inhibitors. We observed that 2 nanomolar and 200 nanomolar trametinib significantly reduced apoptosis and increased average neurite length in glutamate-intoxicated cells. And trametinib treatment significantly increased average neurite length in stress granules-induced model. And we observed that selinexor significantly reduced cell death in both stress-induced models. So in general, our findings suggest that exportin 1 and MEK2 could be auspicious drug targets to be further explored for the treatment of ALS. And with this, I want to thank the whole Maximo team for their great work, and thank you all for your attention.
Thank you very much for this nice presentation about potential new drug targets. Are there some questions? No? Yes.
Hi. Hi. Thank you very much. That was very interesting. I was just curious if you examined how potentially cation channels might be affecting the activation of the ERK pathway, for example.
What?
Cation channels in neurons, for example.
This we haven't checked, but MAP kinase pathway is pretty much big and like a lot of players involved. And of course, it needs to be upstream, downstream of this target needs to also be investigated.
Yeah, I was just curious because as far as the guts, for example, as far as I know, protease receptors are known to activate the ERK signaling pathway. Exactly. And so I was curious if you've looked at something like that as far as the brain is concerned. Thank you very much.